Interphase is separated into three phases, G1, S, and G2. In the G1 phase, new cells grow and make organelles and proteins. In the S phase, chromosomes are replicated and DNA molecules and proteins are made. In G2, organelles and molecules are made. These are then used for division. In prophase, the DNA condenses and the centrioles separate and go to opposite poles. Spindle fibers are also produced, and the nuclear envelope and nucleolus break down. Next, during metaphase, the chromosomes line up in single file towards the center of the cell. Each chromosome then attaches to a spindle fiber on both sides of the centromere. Then in anaphase, the centromere splits apart. Chromatids are then pulled to opposite sides of the cell or the poles. Lastly, in telophase, the DNA uncoils, spindle fibers break down, two nuclear envelopes form around the DNA, and two nucleoluses form in the nuclei. Then the cell membrane pinches in, getting ready for cytokinesis. In cytokinesis, the cell membrane pinches in a little bit more, and the cells slowly begin to separate. Then we end up with two daughter cells that have the same amount of chromosomes as the original parent cell.
Interphase 1 is made up of three parts, G1, S, and G2. In G1, new cells grow and make organelles and proteins. In S, chromosomes are replicated, DNA molecules and proteins are made. And in G2, organelles and molecules for division are made. The chromatin condenses into chromosomes. The centrioles separate and a spindle fiber begins to form. The nuclear envelope then breaks down. Also in prophase 1, each chromosome pairs with its homologous chromosomes. They form a structure called a tetrad. Crossing over is part of prophase 1. Crossing over is when the chromosomes are in a tetrad. They can exchange parts of their chromosomes. Crossing over leads to genetic diversity for an offspring. In metaphase 1, homologous pairs line up together in the middle of the cell. Spindle fibers attach to the chromosomes. In anaphase 1, homologous pairs are separated. Spindle fibers pull chromosomes to opposite sides of the cell. In telophase 1 and cytokinesis, the chromosomes gather at opposite ends of the cell and lose their distinct shapes. Two nuclear envelopes will form. In cytokinesis, the cytoplasm pinches in half, and each daughter cell has a unique set of chromosomes. Meiosis 1 results in two haploid daughter cells, each with half the number of chromosomes of the original cell.
In meiosis 2, the cell splits in half to begin the process. Meiosis 1 results in two haploid daughter cells, each with half the number of chromosomes as the original cell. Also, each cell is haploid, meaning it contains one set of chromosomes. Along with this, the nuclear envelope breaks down. In metaphase 2, the chromosomes line up in single file along the middle of the cell. Along with this, spindle fibers attach to the centromeres. In anaphase 2, sister chromatids separate and move to opposite ends of the cell. In telophase 2 and cytokinesis, the two daughter cells split into 4 haploid cells, and the nuclear envelope reforms. In males, 4 haploid sperm cells are formed. In females, most of the cytoplasm goes into one cell that becomes the egg. One egg and 3 polar bodies are formed. Mitosis is used for growth and repair. It's involved with 2 body cells. The cells are diploid and the genes are identical. Meiosis is for gamete formation. It involves 4 sperm, 1 egg, and 3 polar bodies. Its cells are haploid and its genes are different.